Okay, so now that we've talked about the secondary level of protein structure, we can move on and talk about tertiary structure. So tertiary structure is just the ultimate 3D shape that a polypeptide takes on. So it includes secondary structures, but also other kinds of structures that connect them together. Um, Remember that the primary structure depends or is defined by, in fact, the sequence of amino acids of the protein. The secondary structure are mostly held together by non-covalent interactions between the atoms of the peptide backbone. So it does not depend as much on the amino acid sequence, but tertiary structure does. So tertiary structure involves interactions between side chains and also hydrophobic effects between side chains and water. And so it depends a lot on amino acid sequence, and the tertiary structure can change quite a bit from moment to moment depending on what the protein's binding. And whereas there were only really two major kinds of secondary structure that we talked about, the alpha helix and the beta sheet, there are way more types of tertiary structure. So really it's easiest just to lump them together or classify proteins into basically two major types of tertiary structure. Fibrous proteins and globular proteins. So fibrous proteins are what they sound like. They're just proteins that form long fibers or strands. And then globular proteins are ones that are globular. They're more spherical generally, or just maybe amorphous overall. They don't have really any corresponding shape. Fibrous proteins tend to be structural proteins, so both the structural proteins of inside the cell, but especially extracellular proteins, so the proteins that make up the material that your cells are embedded in your tissues, are mostly made of fibrous proteins. And then the globular proteins do everything else. So enzymes are globular proteins, receptors are globular proteins. Anything sort of non-structural tends to be a globular protein. In fact, we can see, I can show you kind of what they look like, again in 3D. So if I switch to these modes, so you can see our examples here. This is a fibrous protein. This is a protein called keratin, and you can see it mostly consists of these two alpha helical strands. I just sort of showing them here at the molecular surface, so you may not be able to see the helix part, but that's okay. You can see that each individual strand just sort of wraps around each other, and they're nice and elongated. Whereas over here, this is a globular protein, and actually it's an example of an enzyme called pyruvate kinase. But what's important is you can see it has no real particular describable shape. It's just sort of a glob. Now, you may see some proteins where the shape, there may be some combination. So you could have a protein that has a fibrous part and a globular part, but for the most part, especially with enzymes, they're kind of all of one or the other. So we'll start with fibrous proteins first, just show some examples of them and how they work. So in fact, the protein I just showed you was a molecule called keratin. Alpha keratin in particular is an important protein in, again, extracellular tissues. It's also the protein that makes up things like hair and fingernails. So the alpha keratin protein consists of two chain coiled coils of alpha helix. So it's actually two separate polypeptides, both of which form long alpha helical structures, and then those wrap around each other. And then a cluster of those coils together form what's called a protofilament. The protofilaments bundle together into protofibrils, which then 3 or 4 of those together form an intermediate filament. And then each hair cell consists of a whole bunch of bundles of those intermediate filaments. So one hair is actually maybe hundreds of elongated cells with long bundles of these proteins packed in very tightly, and of course with no other cellular components. So a hair cell is really just a dead cell that's now packed with protein, but all the water and sort of other cellular material has been drained out or has deteriorated. And again, the same goes for fingernails and some other body parts. In animals like rhinos, for example, the horn is actually made mostly of keratin protein, for example. So it's a very strong structural protein. That's why it's found in these body parts. So again, this is just a close-up view of the protein. So each individual protein subunit is these two polypeptides that are just wound around each other. And then a bundle of 4 of those make up a protofilament. And then a protofibril is a pair of protofilaments that are then wound around each other. So it's this multiple levels of coiling that also give proteins like keratin and other fibrous proteins their strength, because just like a rope is made up of many tiny strands of thread or yarn that are wrapped around each other, fibrous proteins are built the same way. Another interesting thing worth mentioning about keratin is that the helices, the individual polypeptides, are held together by a lot of disulfide bridges. So remember that a disulfide bridge is a covalent bond between pairs of cysteine residues. So the cysteine residues form these disulfide bridges, and so each individual fiber is stabilized by those bridges. But you can reduce or break these disulfide bridges with a reducing agent. So there are lots of reducing agents that can break these disulfide bridges, and in fact, a lot of sulfur-containing compounds will break these disulfide bridges. So if you've ever gotten a perm or seen someone getting a perm, the chemicals that you put on your hair to make a perm are basically reducing agents that break these disulfide bonds within the keratin fibers of the hair. And what that does is when you break those bonds and then allow the hair to curl, then they eventually reform. So you add either an oxidizing agent or you just let the bonds reform naturally. And when they reform, they don't all form in the same position they started off in. And so instead of being straight, each individual hair is now curled. So that's what causes hair to curl when it gets permed. And of course, the reverse is true. You can straighten out hair by reducing those disulfide bridges and then allowing the bridges to reform while the hairs have been straightened. Of course, eventually the hair regrows and the straightened hair will replace the curly hair. But anyway, that's how those structures are created. Collagen is another really important structural protein, especially for animals. In fact, collagen is only found in animals. Collagen forms this kind of unique structure. Each individual unit of collagen is actually 3 left-handed helices. So again, most alpha helices we talked about are right-handed helices, but collagen fibers are able to form these left-handed helices for a couple reasons. One is their helices contain these combinations of glycine, proline, and hydroxyproline residues. And so simply the geometry of those residues allows the helix to form or the strand to form a left-handed helix. And then the 3 left-handed helices wrap around each other to form, again, kind of a superhelix. So these hydroxyprolines are just modified versions of regular proline. And it turns out that the addition of the hydroxy group to the proline converts it from this configuration, what we call endoproline, where the whole thing is kind of pointed up like this, and into what's called exohydroxyproline. And this shape is what allows or helps the left-handed helix form. Now, one thing to mention, and you can see again in a collagen protein, bundles of these connected to each other, again by disulfide bridges, can form these long fibers, and then those fibers again form a big part of the matrix that your cells are kind of embedded in a lot of tissues. So especially places like skin and other tissues, muscle, the connective tissue of your body has lots of collagen, and it's what all of your cells are kind of stuck into and hanging from and attached to. So it's a huge structural component of your tissues. And like I said, it's actually only found in animals. And it turns out you can take collagen and from animal tissue, dry it out, and grind it up. Actually, you first boil it, and boiling it denatures it. We'll talk about protein denaturing later, but it kind of unfolds it. All of these kind of nice structural elements are separated, and then you dry that out and let it reform in the presence of water, and it will form a gel. In fact, the protein, after it's denatured like that, we call it gelatin. So jello, which is what Jello is made of gelatin. Gelatin is actually an animal product. It comes from usually the skin or sometimes the hooves or bones of cows. So it's actually a beef product. But in any case, the other important thing or interesting thing about collagen is getting back to this modified proline is that the reaction that produces hydroxyproline, and this is of course is what we call a post-translational modification. So that means that the protein is made with regular proline, but then later on at some point before it becomes part of the tissue, some of these prolines are converted to hydroxyprolines, and the reaction that the enzyme that produces or converts proline into hydroxyproline, among other things, requires a molecule called ascorbic acid, or sometimes called vitamin C. So in fact, one of the symptoms of vitamin C deficiency is sort of a gradual degradation of tissue. So you have things like hair falling out, eventually teeth falling out. Long term, it leads to muscle deterioration, bad skin rashes, all kinds of nasty things. And in fact, the original term for vitamin C deficiency, or the disease that it causes, is scurvy. And so scurvy, you probably heard of as the disease that a lot of sailors used to get, and the reason they used to get scurvy, it turns out, we know now, is because on long sea voyages they didn't have a lot of vitamin C. So without, they mostly just ate fish and didn't have a lot of access to fruits and vegetables, and so that's where they would get scurvy. So in fact, once they discovered that vitamin C, or that eating fruits and vegetables would help that condition, it mostly went away. Scurvy occasionally still comes back, though. You still occasionally find people with severe vitamin C deficiency. Usually it's people that just have a generally poor diet. So if you just happen to live in a part of the world where you don't have access to fruits and vegetables, fresh fruits and vegetables, then scurvy can be an issue. It can even be an issue among places where there's plenty of fruits and vegetables available, but sometimes in certain situations people just don't eat them. So in fact, this is not an uncommon problem among college students. So college students sometimes will, if they're just caught up in studying or whatever, will kind of gradually let their diet deteriorate and not eat fruits and vegetables. And so you do see sometimes otherwise healthy people develop scurvy because they do not get enough fruits and vegetables. And then another important example of a fibrous protein is called fibroin, and this is also called silk protein. So some animals, like spiders, for example, will produce this protein and use it as webbing. So they have these silk glands that basically extrude this protein as these long fibers. The fibers kind of tangle up with each other, and then it forms stronger webbing. Now, in this case, though, unlike keratin and collagen, fibroin is made up of proteins that mostly form beta sheets. So a fibroin protein is really stacked anti-parallel beta sheets, and they contain lots of alanine and glycine residues. Those are obviously very small and non-polar residues, so they easily pack together very tightly. They're actually attracted by van der Waals forces, and that allows them to be both very flexible and very strong. So those beta sheets can kind of slide past each other and can twist very easily, but are very tightly compacted and have a lot of strength once they kind of wind around each other. So that's where that property of silk comes from. And there's lots of other examples of again, these fibrous proteins, but mostly they're found either in the extracellular compartment of tissues, where again the cells are kind of embedded, or they're found in parts of the body that need to be extra strong or hard, like hair and horns, and again like silk protein here. So their main job is structural. Globular proteins again are much harder to classify. There are way more types of globular proteins. In fact, there are hypothetically an infinite number of globular proteins out there, or everything that's not fibrous protein basically is considered globular. So these are all just different views of the same globular protein. This is myoglobin. And again, you can see it in 4 different views. Over here, you can really see the secondary structure. So you can see that the alpha helices, where they're positioned, And then this is just the molecular surface, so you can see what the actual protein would look like if you could see it. Again, it has to be able to hold that heme group, so that little pocket there has to be exactly the right shape for that heme to fit in there. This is showing you, again, still the secondary structure with the individual residues sticking out. And then over here, you can see the residues, and they're sort of labeled by their hydrophobicity. So that's why the yellow ones are kind of tucked deep inside the protein, where you can't see many of them because, again, they're exposed to water. So again, for fibrous proteins, their shape is important because it contributes to their structure, and their structure and the shape contributes to their strength. That's their main job, is just to hold their shape and resist force. Globular proteins, meanwhile, their shape is also determined by their sequences, their amino acid sequences, but their shape is important to their function because, again, if you look at a protein like myoglobin, its only job is to hold on to this heme group. And so it's able to do that because the little pocket that forms there is exactly the right size and has attachment points for a heme group. And then also to allow it to attach to and release oxygen in the blood, actually in muscle. And so that's just an example of why the structure of these proteins is so important, because without that structure, it would not be able to do that job. Again, it's harder to classify globular proteins, but we can talk about certain themes or certain patterns that show up. So when you see a common set of secondary structures within a protein or within multiple proteins, sometimes those are called motifs. So these are two examples of motifs. This one over here, this is the beta alpha beta loop, and it's called that because it consists of an alpha helix with two parts of a parallel beta sheet at either end. So the beta sheet consists of two strands. This is the first strand here, and then it loops around to the alpha helix and then back to the other strand on the other side. So that's one example of a motif. This is another one. It's called a beta barrel, where you have a bunch of beta strands all kind of wrapping around a central cylinder. And again, multiple motifs may show up in a single protein. So again, this is that protein pyruvate kinase, and you can see that there are at least three versions of that beta alpha beta loop within the protein. So over here, you can see we're looking at our pyruvate kinase again, and you can see that beta alpha beta loop shows up in several places. So here's one version of here's the alpha, and then again two beta strands coming out of either end of it. And then again, two of those kind of end to end form part of the structure of this protein. So again, those kind of common patterns that show up a lot, we just call motifs. So another way to describe the tertiary structure of globular proteins is by domain. So a domain is really just any sort of independently stable part of a protein, usually that has some sort of functional significance and is connected to the rest of the protein by a relatively small connection. That so this is an example. This is a protein called troponin. Troponin we'll talk about later because it's important for muscle function. But troponin binds to at least two things. It binds to another protein called actin, but it also binds to calcium ions. And this sort of orangish part of the protein over here is called the calcium binding domain. And it's called that because its job is to bind to ions of calcium, but it's called a domain because it's largely separate from the rest of the protein. They're connected, but they're only connected by this kind of narrow neck here, which means that the calcium binding domain is largely separate from the rest of the protein. And that means, among other things, that sometimes you can have, for example, mutations in one domain of a protein that may or may not affect the function of the rest of the protein. Also, you can study protein structure or protein function sometimes just by looking at one domain or the other. So you could actually, for example, generate what's called a chimeric protein by taking the functional domain of one protein and attaching it to, say, the structural or catalytic domain of another protein. and then you can do all kinds of fancy stuff experimentally that way. So again, whether a part of a protein is really a domain or not is kind of subjective, but if you have a region where the structure can be sort of separated hypothetically from the rest of the protein without altering its function, or if it's independently stable without the rest of its protein, then it would be considered a separate domain. And then we have, there are lots of proteins actually that don't really have any describable tertiary structure at all. I mean, they have tertiary structure, but it doesn't really fit into any other classification, or it may change quite a bit or be very highly variable. And so these are called intrinsically disordered proteins, or sometimes intrinsically disordered domains. So you could have a protein where part of it is structured or ordered and part of it's not. And all that means is that the protein is highly variable in its shape, and that protein can change shape depending on what it's binding to or what the environmental situation is. These are especially important for proteins that bind to lots of other things. So this is an example where this protein has this highly variable domain that corresponds to basically both the amino and the C terminus. So here they've actually graphed this, what's called the Ponder score. And basically, it's not important what that means. It's just a relative measure of how disordered a given amino acid in that sequence would be. And so this shows that basically around the amino terminal and around the C terminal, you have lots of disorder, or you're more likely to find disorder, as opposed to the sort of central core of the protein. And that those positions, especially the C terminus, also correspond to the binding domain of lots of other ligands or proteins. So these are all different examples of proteins that or configurations this protein takes on when binding to other ligands. So binding to cyclin A or sirtuin, CBP bromodomain, and so on. So these are each different proteins, and when each of these binds to again the C terminus region of our intrinsically disordered protein, it kind of takes on different shapes. So that's why these intrinsically disordered proteins, it sounds like they maybe would be less useful or less important, but because they have the ability to vary their shape quite a bit, they're important for a lot of these binding properties. We'll talk about protein binding more in the next chapter too. And then quaternary structure refers to just the assembly of two or more polypeptides into a larger complex. And there's lots of examples of quaternary structure. Again, some proteins only exist with multiple subunits. So this is a molecule of hemoglobin. So hemoglobin is again the protein that carries around oxygen in your blood. But a single hemoglobin protein is actually 4 separate polypeptide subunits. So a subunit of a complex like this, a protein with quaternary structure, is just a single polypeptide. And it turns out that the hemoglobin has 2 types of subunit that are different, so alpha and beta. And so those are 2 separate polypeptide sequences. They each have their own DNA sequence. They're each translated separately. But then they form this larger complex. So 2 alpha subunits and 2 beta subunits come together to form the whole quaternary structure. Each individual set of 1 alpha and 1 beta is called the alpha beta protomer. So really a hemoglobin consists of 2 alpha beta protomers. But again, that's just an example. So there are other proteins where all of the subunits are exactly the same. You may see proteins with quaternary structure where they consist of many different subunits that all are completely different from each other. Or you may see quaternary structure that is just temporary. So you may have 2 subunits that only assemble under certain circumstances, and then they separate in response to some signal. And again, this quaternary structure plus the secondary other types of tertiary structure that we talked about are likely to change with the condition the protein's in. And in fact, the ability of these structures to change is really important for protein structure. So we'll talk about that more in the next chapter too. Now, how do you determine protein structure? There are lots of ways to figure out a protein structure. nowadays a lot of protein structure, or some of it, is worked out kind of mathematically. So if you know an amino acid sequence, you can use mathematical models or just a simulation to kind of fold that protein up and try to find what its structure is likely to be. But to empirically determine protein structure is a little bit trickier. Again, some proteins are big enough that you can kind of see them directly with the right type of microscope, but the detail is not very high. So if you actually want to figure out the detailed structure of a protein, you have to use some pretty advanced, sophisticated techniques. One thing you can do is called circular dichroism spectroscopy. So what that means is you're using circularly polarized light to visualize a molecule, and or to just like you would with other kinds of spectroscopy. So if you shine a circularly polarized light beam through a protein solution, you can modify or rotate that polarization, and the degree of secondary structure in that protein will actually affect this property. So proteins with a lot of alpha helices will give a different spectrum than those with a lot of beta conformations, and then those with a lot of random coil, meaning not a lot of secondary structure at all, will give a totally different spectrum as well. So that's one way to kind of roughly determine the degree of secondary structure in a protein. Again, if you want more detail than that, you need to use more advanced techniques. So the two most commonly used ways of directly determining the structure of a protein are X-ray crystallography and NMR spectroscopy. So X-ray crystallography has been around a long time. It was probably the earliest method of determining protein structure and the structure of other large molecules. So X-ray crystallography has been around for decades, and it basically involves pointing a beam of X-rays at a crystallized sample of a protein, and then letting those X-rays kind of diffract. And then so when a light beam hits a certain surface, it will diffract. And then if you put an X-ray film behind your protein sample, you will see little dots on that X-ray film that correspond to the diffraction pattern. So this picture here is an example of a diffraction pattern made by a protein. And again, this basically, you're still kind of using light to image a protein, just like you would with visible light. But one reason you can't look at a protein under a microscope is that they're so small that you're down to where the wavelength of visible light is too long to really reflect and interact with proteins. But X-rays have shorter wavelengths, and so they're able to interact with these molecules better. You can't really make a microscope out of X-rays, so instead we just use this diffraction pattern. And then basically the diffraction pattern, nowadays you would feed it into a computer. In the old days, you would have to do a lot of really complicated math to figure out, based on the diffraction pattern, the angles that the light were scattered at. You would have to go back and try to make some guesses about the structure of the protein. But X-ray crystallography now is much more sophisticated in terms of its ability to determine structure. There's some limits with X-ray crystallography. For one thing, your protein has to be able to crystallize. So that means that it has to form a regular crystal. You have to purify the protein and allow the proteins to clump together in a very regular, ordered way for X-ray crystallography to work. And there are a lot of proteins that just don't do that. They don't crystallize easily, and even if they do, you have to have them in large quantities for them to do that in a useful way. But X-ray crystallography is nice because it works on really big proteins. In fact, there's no limit to the size that you can use X-ray crystallography on. But it works less well on certain types of proteins, like membrane proteins, for example. NMR spectroscopy is a little more complicated. Basically, NMR stands for nuclear magnetic resonance, and what you're looking at is basically the properties of the nuclei of individual atoms within a molecule. And so, without getting too far into the physics, every nucleus of every atom can have a magnetic moment to them, and that magnetic dipole can be affected by adjacent atoms. And so an NMR spectroscopic machine will basically tell you about the properties of the atoms in your molecule if it's been purified. And then again, using a computer or using complicated math, you can figure out the structure of even big complex molecules using this technique. Now with NMR spectroscopy, it has the advantage of you don't need to crystallize your protein, but again, there are certain proteins, especially large proteins, that NMR spectroscopy is not very useful for. So these are just how these structures are determined in the first place. So whenever you see a three-dimensional picture or cartoon of a protein, it was worked out probably by either X-ray crystallography or NMR spectroscopy. But again, you can also kind of work out these structures mathematically with computer programs as well, just from the amino acid sequence. All right, so next time we will talk about some of the processes that cause folding and degradation of proteins.